Hey everybody, it's Ms. Hunt, and today is Wednesday the 2nd. I hope you're doing great and ready to get going for biology. So I'm trying to do Wednesday, Thursday, and Friday in this video. So if you are at home, then take your time and pace yourself for each of those days, okay? On Wednesday, we did our notes for chapter 10 and our study guide on meiosis. We are just doing section 1. So let me run through some notes real quick with you for meiosis. Okay, we're going to start from here. Okay, and on the classroom board, I put up a little list of the difference between mitosis and meiosis. Okay, so hopefully I can put something up for you guys too, so you can be able to do it. In mitosis, we said we had daughter cells were identical. We said we have 2 new cells. Okay. All right, so this is what we said for mitosis. We have daughter cells that are identical. We get 2 new cells, and interphase is G1S, G2, mitosis and cytokinesis. That was the cell cycle for mitosis. We're going to compare that to meiosis and see how it's different and how the process this is. So first of all, meiosis sounds a lot like mitosis, and especially when we're Southern, it sounds a lot alike. So I usually emphasize the E in meiosis, even though it's not correct. So when you go to college, don't say it's meiosis because your professor will laugh at you, okay? So I'm just emphasizing it so that you can understand me. So meiosis also causes us to put a lot of little mnemonic devices, okay? Okay, so in meiosis, we're making a cell, just like in mitosis and the cell cycle, we're making a cell. This cell cycle involves meiosis instead of mitosis. And in this cell cycle, we're making a different cell. We're making our gametes. Gametes are egg and sperm cells. So we're making our reproductive cells. And reproductive cells have half the number of chromosomes. So this gives us some different things that we have to do. We oftentimes think about meiosis, and I'll say to think about it as a gametes. And I always say gametes meet. That's egg and sperm. So if you kind of think about meiosis like this, it kind of makes you think, oh yeah, that's what we're talking about. And even if you wrote sex at the bottom of it like that, then you're thinking this is a sexual reproduction, because next week we're talking about asexual versus sexual. So this is a sexual reproduction where we make the egg and sperm and we make those gametes. So there's lots of things we can think about with meiosis that's involved here, okay? All right, so the human body has 46 chromosomes, and you get 23 from each of your parents. We call these homologous chromosomes. You might want to underline HOMO, homo in Latin. That means same. So these are the same chromosomes, one from each pair of your parents. Homologous chromosomes have the same length, they have the same centromere position, and they carry the same genes for a trait. So let's say this top trait right here was like how to build a heart. This is the blue one, so let's just say that that's Dad's information on how to build a heart, and this is your mom's information on how to build a heart. And you have each one of these in every nucleus of every cell in every part of your body. Now, which one your body reads, well, that's genetics for dominant, recessive, and those kind of things. But right now, you have both. In your reproductive cells, one of these is going into one reproductive cell and another one is going into a separate one. That way you're giving half of it, but you're still giving information on how to build a heart. These are called homologous chromosomes. So let's take a look at real life chromosomes. This is called a karyotype. It is a picture of the chromosomes from a human body. So scientists take the nucleus, they take out the chromosomes. It involves putting them on a special kind of piece of tile. It's called a gel plate, but you put them on a tile and you add a little bit of electricity and they stretch across. And then you take a picture of them using a special computer and then you cut them out and you pair them up. It's a very complicated process, okay? But this is what they look like when they come out. So these are actual real chromosomes instead of these little nice cartoon bubbles, okay? So right here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, all the way down here to 44, 45, and 46. So you have 46 individual chromosomes. We often put them in 23 pairs of chromosomes. So these two are the same length. They have the same genes, and you can see those little dark patterns are different proteins that they're coding for inside of here. Remember, in a chromosome is a piece of DNA wound up really tight. So these are different patterns that are being in there, and they have the same centromere position. There's a little pinching going on right there. That's the centromere. So these are what we call homologous. So they're going to double, and when they double, that's when you get the little X's. When these double, you'll see 2 here, and you'll also see 2 here of the X's. So this is a human being. It's got 46 chromosomes. And is this genetically a boy or genetically a girl? You can tell by the chromosomes. All right, right here, this is the 23rd pair of chromosomes. This person's inherited 2 of the X chromosomes, so this is a girl. All right, this right here is a boy. All the chromosomes are the same, and my picture's a little blurry, but it shouldn't be that blurry, but it is. And right down here, we have one X and then one Y, and the Y is little one, so that is a boy. Okay, we can also see when there's genetic problems. Right here, I don't know where to put my face, okay? We have a person who has a problem, okay? I'll put me right here. All right, the one chromosome is the same, 2, 3, 4 pairs, 5, but right here, they have 3 of chromosome 21. Anybody know what that's called? If you guess Down syndrome, then you're right. This person has Down syndrome. And this person is a girl. She's got 2 X chromosomes. This is a female with Down syndrome. Remember, that'll be in every chromosome, in every nuclei of every cell in her body will have Down syndrome. So there's nothing that can be treated about it because it's in every cell of every part of her body. Okay. When we do meiosis, it's called a reduction cycle because we're going from haploid, which is half the number of chromosomes, to a diploid, which is a full number. Sorry, actually, we're going from a diploid to a haploid. Okay. An organism produces gametes to maintain the number of chromosomes from each generation to the next generation. Human gametes contain 23 chromosomes. So remember, gametes are your eggs and sperm, and they're going to have half the number of your chromosomes. A cell with n number of chromosomes is called haploid cells. So underneath here, write the word half or haploid, half the number of chromosomes. And we use the variable n to represent this. And I know you're like, Ms. Hunt, please don't do variables in science, but that's what we use is n for half the number of chromosomes. A diploid contains 2n, double the number of chromosomes. So we have 23 haploid, which makes 46 diploid. So what if I said a turtle had 20 diploid chromosomes? What would be its haploid? And you would say, well, 10. What if I said a fruit fly had 8 N haploid? What is its diploid? You would say 16. So you have to be able to go back and forth on that between the full set and then the half set. All right, meiosis is going to go meiosis 1 and then meiosis 2. So we go through it twice. Meiosis involves us going from diploid to N, being adults, reproducing. Now, these two people probably have gone to college, gotten married, and chose to have kids. And now they've reproduced. The egg from the female is haploid, and the male sperm is haploid. And together they're going to fertilize and make a zygote, which is the first cell that's diploid, and that's 2N. And it goes back to an adult who's diploid. So this cycle goes through your life. All right, stages of meiosis 1. We reduce the number of chromosomes by half through separation of homologous chromosomes. All right, it involves 2 consecutive divisions, meiosis 1 and meiosis 2. All right, first of all, we have interphase. So our cell's just being a cell. It's growing, it's preparing to divide. It also does a very special S phase, very special S phase. What happens in the S phase? Do you remember? Should tell you the DNA synthesizes and doubles. Okay, prophase 1, pairing of homologous chromosomes. So remember, this is dad's and this is mom's. They've doubled in the S phase, and now they're pairing up. Okay. Prophase 1 has something very special called crossing over. Crossing over is when they exchange a piece of DNA. So the chromosome switches. So down here, you probably can't see it because of that little sign right here. Maybe I can, there we go, I can move it. Okay, there we go. You see that these 2 homologous chromosomes switch a piece. So if a child inherits this gene, he'll inherit most of the dad's information, but a little bit of the mom's information right there. All right, what crossing over does is it gives variation to a species. It makes sure that you are not inheriting all of the male or female's information, that there is some variation. All right, so that was prophase 1. Okay, prophase 1, pairing up, nucleus starts to disappear. That's just like mitosis, right? It's the same thing, except it's a little bit different. All right, we did crossing over. All right, metaphase 1 looks a little different. All right, normally metaphase in mitosis had all the X's on the equator line, but this lines them up by homologous chromosomes. So we're going to be separating homologous chromosomes. We have 2 X's on that equator line. And then anaphase 1, this looks different. The entire sister chromatids are pulling to each side. So homologous chromosomes separate. So you might want to underline or highlight homologous chromosomes. If we're talking about homologous chromosomes, we're talking about meiosis 1. So this is anaphase 1. Telophase 1, nucleus starts to come apart. Okay, we're going to have cytokinesis after this. The notes don't show it, but it does do cytokinesis, and it immediately goes into prophase 2. Okay, for prophase 2, same thing. The nucleus starts to fall apart, spindle fibers start to form. Metaphase, now this looks a lot like mitosis. The chromosomes are lined up, the sister chromatids are lined up on the equator. But notice we have 2 cells here, not 1 cell. We're not 1 cell going into 2, we're 2 cells going into 4. All right, now we're pulling the sister chromatids apart. Make sure you underline that sister, okay? Sister chromatids, this is meiosis 2, and this is what it looks like. They anaphase away, sister chromatids are being pulled apart. Notice you can see where the crossing over happened from prophase 1. All right. In telophase 2, nucleus starts to reappear, and we have cytokinesis. We have the pinching apart. These cells are N, meaning half the number of chromosomes. They are haploid. All right, the importance of meiosis. Meiosis is 2 divisions. We produce 4 haploid daughter cells that are not identical to the parent. It results in genetic variation. Meiosis provides variation depending upon how chromosomes line up at the equator. 4 gametes with 4 different combinations and 4 chromosomes can result. So how they line up here, is it mom, where are they at, who goes to one side, and where crossing over happens. All of that can give us variation. And then which one of these eggs is the one that gets fertilized, or which one of the sperm gets fertilized. It's all a lot of randomness to give us variation. Okay. All right, sexual versus asexual is the last slide here, and that's what we're going to be talking about next week. Remember, asexual, the organism inherits all the chromosomes from the single parent, and they're genetically identical. So in asexual, all they're doing is mitosis. But in sexual reproduction, we're doing meiosis. The benefit is the genes multiply faster over time. We can have evolution, and we can have variations in our species. All right, so I'm going back to my chart real quick and looking at this. So we said daughter cells identical and 2 new cells. So what do we put over here? The daughter cells are not identical. And we have how many new cells? 4. 4 new cells, okay? So 4 new cells, daughter cells are not identical. All right, our phases, we said interphase, mitosis, cytokinesis. Over here, we still do interphase. We still do G1, S, and G2, but then we do meiosis 1, sorry, I can't write and talk at the same time, okay? All right, meiosis 1, cytokinesis, and then meiosis 2, cytokinesis. All right, so that's a very quick little chart of the 2 main differences here. Okay, now you have a study guide to do. Okay, after you finish that study guide, all you got to do is section 1, just the study guide. All right, that is Wednesdays. Okay. Did I exit? No, I didn't. Okay. All right, making sure I didn't mess up. I'm going to stop this recording and I'm going to record another one for Thursday, Friday.